we search for Orange machine learning. Can you see my screen? So if we go to Orange Machine Learning, this is where we get this software from. And this is quite a nice piece of software. It's a visual tool. And it means that we don't rely on, if you're not able to program in Python or the such, then you can still conduct machine learning assessments. And this is kind of creating a visual interface, and many of these tools are built and developed in Python. Now, the nice thing about Orange is that it has many video tutorials. And so if we look at workshops, then there are online workshops as well, but there are text help, there is video help, there are video tutorials on this. So it's quite a useful technique to start learning about machine learning and some of the methods that can be used there. So I'll show you an example of this, and I'll build an application. So I have here a blank workbench for Orange. So as you can see from this, there are a number of tool sets that we can use. There are tool sets which you can download and incorporate for things like spectroscopy, bioinformatics, geographic data, single cell data. There are also, when it says bioinformatics, we can pull in data from things like gene expression omnibus, or we can basically incorporate database methodologies. So that there are a lot that we can do text mining, for example, as well. So there are a lot of potential tools which we can utilize here to achieve this. So what I'm going to do, first of all, we need to load a file. We need to load in some data to work with. And this system uses comma-separated value files, CSV files. So I'm going to create a node called file, and I've just literally dragged that from our workbench here. If I want help on this, I right-click on it, and I click help. And it tells me all about what I have to do to load data and format data and the like. So that's the first bit. If I double click on it, you can see I can, well, I've already selected a file here, but I can get information about the file that I've selected. So if I've double clicked on it, I can, if I click here, I can look at the data for the course. I can browse my computer to find data, and I can reload data. So that allows me to effectively set the data I'm looking for. What's happening? Sorry. Now, once I have that, I maybe want to select what I'm going to use. So I want to say what are my input values and what is my target value. So the way I do that is if I use the select columns tool. And that allows me to select from my data, select columns, or particular columns of interest. So if I click help, you can see how it shows me how to use that, and gives me details and examples. Now, in order to use that, I need to create a link between my file and the columns. So the way I do that is I move my mouse over the dotted line here and drag, and that creates a link between those two. And you can see that what it's doing is feeding data from the file into the select columns function. I can click on that and you can see it's going from data to data. So now when I double click on that, you can see I have features. So these are the things I'm using for my values. And now you can see I've got disease POS-neg in here. Well, I don't want those in there because those are my independent variables. And so I keep those in the features, and I want to put a target variable into target. So I want to predict here disease, yes or no. So I close that and just check that it's saved. Yeah, that's saved. So there we go. Right, I'm just going to take that away from full screen because that is being problematic. When the tools become full screen, it's hard to see how they fit in. So now I have effectively a data set that's been read in, and I have selected the things I want to use as dependent and independent values, and in the select columns. So maybe I want to look at the structure of the data. So if I go to unsupervised, I can do a principal components analysis. So here we have PCA. I'll select that, and I want to take my data from here into PCA. And so I'm looking at data to data again. And if I double click on this, you can see it's immediately done a principal components analysis. And you can see it tells me the cumulative variance and the component variance at for my data. Excuse me. And you can see here with one principal component, we're explaining, just drag this, one principal component explaining 44%, two components 68%, three components 84%, four components 96% of the variation. And what I select here will be taken forward for further analysis. So I'm going to select 3 for the moment because that allows me to visualize in 3 dimensions. So I want to take the output of this into a data table because I want to be able to look at it in a table. So if again I select the output of that and put that into a data table, you can see I've now got the data for my principal components. And I've also got the variables that are in the data as well. So now I can do something with those. So I can, for example, scatter plot those. So if I click visualize and then take a scatter plot, I drag from one to the other. Now you see here, it's come up with a dotted line. And that's something to bear in mind. When you get a dotted line, it's suggesting that there's an error or something needs sorting. So if I click on the dotted line, you can see now I've got selected data to data. But what I want to do is go from data to data. So if I click data to data, so I click on data, hold my mouse down and drag, and then let go on data, then that fixes that problem. The lines are now not dotted. And from my principal components, you can see where now I've got the separation of the two classes, which isn't very good, I'm afraid. here in the first two principal components. So that is a way allowing us to visualize the data and allowing us to visualize the first two components of that. We can do things like looking for informative predictions and score plots, but I'm not going to go into that here. But we can also take those elements and we can change the features of the graph. So that might tell us what's happening in the first two principal components, but we can also do a linear projection. So we can do a 3D plot, and that has the same principle. You see again the line has become dotted, so I want to go from data to data. Oops. Click OK, and now you can see I've got a 3D plot of the data, and I can move that around. I'm not sure if I can rotate it. But you see here I've selected the first three principal components to plot, and the only problem I think is that I would like to be able to rotate this, but I'm not sure how to do that. If you see right at the bottom here, I have a few tools. So for example, I can save this picture as an image. I can report, I can set visual settings, and it tells me the number of parameters that are used in that, number of and replicates and instances. So you can see that, that I can quickly generate with a simple workflow a principal components analysis. That's not as such machine learning, it's a way of visualizing data. So I want to look at some alternative methodologies. So another form of an assessment of data is, if we go to unsupervised, is a clustering, hierarchical clustering. So to generate a hierarchical clustering, I need a distance matrix between every point in my population. And you can see here by clicking on distances, I can generate that, and I can use a range of different measures to calculate that distance. I can also select based on rows and columns. So for example, it may be that I want to look at this clustering of parameters such as proteins, or I maybe want to look at the clustering of individuals. So I'm going to stick with rows and Euclidean distance as a standard at the minute. But again, so if I drag from the data here to distances, if I click on it, I don't see anything, but then I need to use those distances and I can use them for various things like k-means, correspondence analysis, but I'm going to use them for hierarchical clustering here. And if I drag from here, so you can see the data feeds into the distance matrix, and then the distances come from there to the clustering. You can see now that I have generated a hierarchical clustering map. I've used complete linkage here. I want to zoom out. I'll maximize that to show it. And you can see now that I've got my hierarchical cluster dendrogram, and I can click on individual bits and it will give me and highlight certain clusters. So for example, that. So this allows me to effectively select individual cases as well. So that's doing the hierarchical clustering, but that's also not a form of machine learning. So what I'm going to do is I'm going to take another form of machine learning, and I'll take to start with logistic regression. So if I click on model, you can see I have various options for modeling data. I have some common machine learning methods. So for example, I have tree methods, I have random forest methods, I have support vector machines, I have linear regression, logistic regression, naive Bayes, AdaBoost, neural networks. Now, many times we get asked the question, well, why didn't you use this method or that method? And it can be quite a hassle to run a separate method, to code a separate method, and then try that out for your particular data set. Whereas what we can do here is quite quickly generate a workflow that tests different methods. So for example, if I take a logistic regression, and I'm going to take the data from here, then I immediately can run a logistic regression on the parameters I set. And but I will need to evaluate that. So if I click on evaluate, you can see there is a tool which is called test and score. So if I take the output from my logistic regression and take that to test and score, but also I need to take my data because I need to tell it what I'm scoring, then immediately by clicking on that, you can see that I am able to test the logistic regression method, and I immediately get an area under a ROC curve, and I get a precision and a recall value. Now I can do various things in here like cross-validation, leave one out, training data, test data, selecting the target class, looking at model comparisons. So I can do quite a lot with this, but it's fed, the key point here is it's fed into a workflow that I've generated on the screen. Now something else I can do is I maybe want to visualize the ROC curves I've generated. So if I do that, I can take our results from test and score, and you can see it tags those out as evaluation results and puts them into a ROC curve. So you can see here, although it's not particularly good, I've got a receiver operated characteristic curve here, and that's a good way of testing the performance of a classifier. What we want here, ideally, is an area under the ROC curve of as close to 1 as possible. Anything with a value of 0.8 or more is possible, although 0.7 is sort of acceptable. So that it very quickly allows me to produce a ROC curve for our logistic regression. Now maybe I want to evaluate, excuse me, maybe I want to evaluate some other models, other methods. So for example, I'll drag in an artificial neural network. So I'll take the same data, feed it into the network, and you can see it's got a data tag here. I'll take the output from the neural network, and you can see it's tagged that as a learner, and it's fed into test and score and fed into ROC analysis. So now if I click on test and score, you can see I've got a performance of the neural network, which for this data isn't very good, and I've got precision and recall. And I can compare that with the logistic regression. So if I look at the ROC curves now, you can see that I get a ROC curve, and actually there's probably not that much difference between these. So for this case, we've now looked at logistic regression and neural networks and tested the results and produced a ROC analysis. So let's try something else. Let's try a support vector machine. So a support vector machine tries to find a vector that separates out our two classes through hyperdimensional space. And that vector can be linear, it can be polynomial, it can be a radial basis function, which is a sphere, and we can select that in this. So for example, now if I drag the values from that, we have data coming to our support vector machine, and by clicking on this, you can see I can select the kernel, which is whether it's linear, polynomial, radial basis function, or sigmoidal. And from that, I can then test that model in our test and score. And you can see the SVM actually does pretty well on this data. If I look at the evaluation results, you can see that for this data, a ROC curve is much better. And I think we have a value of 0.6 there. Let's just increase the size. So we can get the area from here from test and score, and you can see the area under the curve is that there was 0.786. So we'll also try a random forest as well. So we can, in our random forest, which uses a number of decision trees, we can effectively, it's an extension of cluster analysis, which allows us to decide which branch a particular individual class belongs on. And we can develop rules that build that, but then with a random forest, we build multiple trees that randomize the data and see where we get. So you can see here we've got 10 trees. I'll bump that up to 20. You can select the tributes in each split, and you can set that you. You can also prune the tree, so you can set the depth of a tree and limit that. So from the random forest method, we now can see that the random forest is doing much better for this data than the support vector machine. If I look at the area under the ROC curve, this is the random forest, and that does better than the SVM as well. So having told you in my lecture that neural networks are a good way to learn, I've actually just demonstrated for this data, which is a very small and artificial data set, that actually random forests are better. But from experience, when I'm looking at high dimensional omics data, then the neural network method comes out better there, because it can cope with the complexity of that, whereas this is a very simple demonstrator. So please don't think that these results are reflective of every situation. But what it does demonstrate is quite quickly, and it's taken me about 20 minutes to do this, I can test a range of different algorithms side by side data set, and I've not needed to program anything here. I've not needed to hard code anything, and just by simple tweaking of different features, then I've been able to test that out. I'm just going to add another approach, which is naive Bayes. So this uses a probabilistic framework to predict whether adding something has a benefit to the model, and from that working out whether probabilistically a feature is contributing to the class. So that gives me another performance measure, and you can see naive Bayes doesn't quite do as well here as the other. And again, from the rock curve, I can see where that fits. So I've now got these 5 methods, and I want to see whether these 5 methods are better on their own, or whether if we take effectively a vote from each one of those, whether we can identify if the collective result is better than an individual result. So I do this by a process called stacking. So what stacking does is stacking receives information from all of the methods. And yeah, learners to learners, that's good. Okay. And effectively combines them via voting. So in other words, if 4 out of 5 of these methods in this ensemble actually say it's this thing, then it will go with that. So it goes majority. So it's quite a simple tool. All we call it is stack. There's no settings as such, but it does do that combination. So if I now take the stacked value to test and score, it takes a bit longer, as you can see, but I now have a combination result. So you can see now actually that the stack doesn't do as well as the random forest. So that suggests that there might be some conflict between the other methods here and the random forest. Normally we would expect the stack to do better, but in this case the random forest has done better. So if you like, I think there might well be a conflict between those features. I'm just looking at model comparison by AUC. It's supposedly showing a table. So interestingly, if I look at testing on the training data, then the net random forest is more or less 100%. If I look on test data, I haven't set that out yet. So let's do a random sampling. Cross validation, test data. No. Yeah, so I've only really got a training data set here that I've selected at the moment. I think I probably have to go to my individual classifiers and set those, or select it at this point. But the point I'm suggesting here is that we've developed this workflow quite quickly, and we are able to test different approaches. Now, this doesn't parameterize in any way as such. It doesn't allow us to select combinations. But you see what I could do here. So this, where I click on select columns, I've got a particular set of inputs in our data that are of interest. Now, so my computer's working hard because I've changed some parameters here. So it's running the updated settings that I've selected, which I think is a cross-validation set. But what I could, just ignoring that for a second, what I can do here is if I click select columns, and I now select It's not doing it because it's running. But what I can now do is in select columns change the parameter set I put in, and I can create multiples of select columns. So I just need it to finish this analysis. So we're on 94% now, so.
It shows, how much time it can take with really big data, right? If we have.
Yeah, that was
really
this is only a small number of parameters. So actually using that, and with now I've stratified them for a cross-validation set, you can see that the stack actually now does better. So this is better because it's generalizing the model, if you like. So what I've done here is I've created now multiple column selections. So for example, I want to remove that. I want to remove that. Maybe I just want to try out 3 different parameters and to predict disease positive negative, or 2 different parameters to predict disease positive negative. So I've now got that in my select columns, and by dragging from this one, it's now removed the other ones because it can't have 2 different conflicting data sets. And now I can see, okay, well, how does that affect my performance? Just check that we're still running that. Yeah, so it's run it, it's stacked it, and you can see now that actually by using fewer parameters in this case, our model isn't doing as well. And if we look at test and score, you can see that the performances have dropped. But you see, by setting up different repeat experiments, where I use different things, then I can prototype a different combination of parameters and try those out. So that allows me to quickly identify different combinations. So there are also many other tools in here. This is just a simple combination of to show some machine learning, but you can see things like we've got data samplers in here, so that will randomly select a subset of data. So you can see I've got a cross-validation approach in here. I can bootstrap and randomize. I can do things like transposing data, which might be important in certain situations. I can use feature statistics, so I can get things like a summary of all the parameters that I have. So if I take, for example, that, I can now get, I'm not sure why that's not too enough. So you can see I can look at the distribution of my different different individuals in the population, and you can see, I can see how they're split between positive and negative. So that's quite a useful visualization tool. I can see the centroids, the dispersion, the minimum, maximum, the percentage missing. So that's quite a useful tool to conduct around the analysis that we're doing. I can check for outliers. So if I drag that there, then I can look at outliers based on here on the Euclidean distance. And if I take that into a data table, outliers. You can see, actually I should take that one away. So if I take those two into here, inliers, outliers, okay, then I should be able to see and I don't think I have any outliers. 24 instances, 5 features. Okay. So I'm just going to get rid of this. And just look at the outliers. So what it will do is theoretically put the outliers into so it's put the outliers into a table. If I create another table from here, I can create a table with the inliers, and you can see these, there's a lot more here, but immediately then this method has cleaned up my data. I've removed the outliers from the data based upon the Euclidean measure. So these are the things which don't work quite as well. So if I take the inliers and put those into the logistic regression, hold on. So I need to take those into the selected columns. So I'm just clearing these ones out. So I'm going to take the inliers, the correct data, if you like, and I will put that into select columns. Let me do that. Hold on. Okay. Okay. I know why. Sorry, I've deleted the file. That needs to go from there to there. That's better. So that's thinking about it temporarily. So what I need to do, so I've got my file. The data comes to select columns. I'm now looking at outliers, and that's the inliers. So I want to use those, and I want to delete that one. So what I've done here effectively is cleaned up the data, and now I can hopefully So this is a little bit experimental, but right. So actually, yeah, the file goes to there. That goes to there. I see. I think I might need, and this was just a trial, but I think I might need to do it that way. With me. Yes, I think that's the right order. Okay. Data. Right, so doesn't it like that? So that seems to, it might be because I'm still running the testing, but it's taking a little time there, but I just need to wait for that to finish while I run this cleanup of the data. I think we're getting there. Right, yeah, that seems to be working now. Okay. Yeah. I don't think it liked it because I was running simultaneously the analysis. So now what I've done with this outliers approach, it's taking a time to run, is I've taken my file, I've cleaned up the data, I've selected the parameter I'm interested in, I've identified outliers in those parameters, and that uses a Euclidean distance measure, effectively a clustering, to pull out things that are not sitting in the central point of the data. And once I have that, I've then fed that cleaned data into the different methods, and it's now running that assessment of the data in order to see if we can improve the model. And it's working through its way through that, and you can see now that it's taking a little bit of time, but we can see how that affects the model performance once we've done that. So again, this is another set of tools here. If I just move to visualize, you can see that we've got many different ways of visualizing. We've got tree viewers, box plots, scatter plots, heat maps. So we can, for example, see by taking from our hierarchical clustering whether we can generate a heat map from the clustering. In model, this is our machine learning supervised approaches, if you like, or our modeling approaches. And in this, you can see a range of tools, but we can also save our model and load our model. So if we find that something's working particularly well, we can, save that model and load it at a later date, or in fact load it into another part of a workflow. So evaluate, this allows us to test, predict, generate confusion matrices, do ROC analyses, lift curves, calibration plots. So for example, confusion matrix, I'll put there, and we'll drag the results out of that once we have our model to see how we get on. We're nearly finished with the modeling. So, for example, now if I look at test and score, not sure if it's any better. I'd need to compare between the two. That's quicker. So this is marginally better, I think. I'll look at what I can do with this is if I look at the confusion matrix, I can see where it's got it right and where it's got it wrong. And again, this model isn't doing particularly well. So you can see 626 were correctly predicted as 0, but you can see 222 were false positives. So they were, false negatives. So they are actually a 1 disease, but they were predicted as a 0. So this is suggesting that really that this data isn't particularly good, or our models aren't particularly good. We can, if we want the instances of that, we theoretically, I thought it said it, we could do this. We could select the ones that are correct. But maybe if we go to data table, for example, we can take the results. And we want selected data, okay. So I think we can then see the results from our analysis in this table, and which ones are zeros and which ones are ones from the different methods. I think these are just selected as zeros at the minute. So if I click on this and select these ones, then that should show me, yeah, these have been predicted as zeros, but we know they're actually ones. So we can see which ones it gets it right for and which ones it gets it wrong for. Okay, so we've demonstrated a number of methods. The cleanup I need to look into to see whether it works, but in theory we should be able to pull data from that and clean up. We've demonstrated that this data is particularly bad, so that's what it is. Other things we can do, there are various visualizations, there are various modeling tools. We can evaluate our models. We have a number of unsupervised approaches, so we've got things like self-organizing maps, which is a machine learning form of clustering. We can prototype our model. So we can look at more detail and do some prototyping, work out how our model is performing in different positions. We can use image analytics. So, for example, we can bring in images and load images and datify images in order to run them through these processes. We can do time series analysis. So we can do things like recursive neural networks. We can look at interpolation of values on the time course. We can do transformations. Textable is something quite new in this, but this is quite interesting if people are interested in text mining. And you can see there are various ways of loading data in, extracting data, tagging data, extracting terms, and so on. So textable is kind of a pre-treatment, and then we can take that into text mining and actually start to do things like pulling out things like sentiment, or we can pull out understanding, if you like, through Corpus Viewer, the text, information from the text. What's also relevant to biologists is increasingly we have single cell data sets. And from here, you can see that we can identify the data set to use. So we can start to do single cell data mining, single cell machine learning by pulling in particular data sets. And, you know, there's, for example, some quite rich data sets here with 8,500 pancreatic cells in humans. So yeah, this is quite powerful. We can start to explore data, and we may not have our own data to analyze, but we can look at data and import data from the sources of others. We can start to generate networks. So some of the network inference stuff I was showing, we can import that here. So we select a network file. I think it's imports as a CSV. I haven't explored this technique because it's relatively new. But essentially, if we have a matrix of cause interaction effect or source interaction target, then it allows us to, that's the problem. It allows us to effectively start to map networks out. Geodata is things like geocoding, mapping. Educational is effectively training. So this is shows visualizations of how we can train different data sets, and it shows how those data sets train. Bioinformatics allows us to do things like identify gene set enrichment. We can load geodata. We can load KEGG pathways here, do volcano plots, do effectively clustering, but from a more biological perspective, so clustering based on gene terms. Associate is relatively new. We can look at association rules. And spectroscopy is something which might be quite relevant to the group here. We can start to do things like we can input data and look at hyperspectra, spectra interpolation, pre-processing, integration of spectra. And so we can do some machine learning pre-treatments of spectral data and then feed that into machine learning tools. So firstly, for a free piece of software, this is very powerful and comprehensive, and doesn't require a lot of technical expertise to get going. One other thing which is important here is, I just mentioned this, is we can actually write Python scripts. And if I drag that in, then you can see we can write Python code into here, or it could be modules of Python, effectively functions in Python, and then we can run those. So not only do we have the opportunity of writing and developing things using a visual function, as we have here, but we can also actually write our own code and incorporate it into our workflows as well. So maybe we have an idea for a new algorithm. Well, what we could do is actually put that Python script down here, and as long as it handles the inputs and outputs properly, could run that in parallel. So there's a lot of potential for further development as well. One of the things I believe we can do, but I haven't figured out yet, is actually we can get to the code behind each one of these nodes as well. So we can get the Python code. That's the basis of each function. And that can also be very useful. So just as an overview, I think this is an readily available tool, easily accessible without large amounts of computational or without large amounts of coding knowledge. And for people who are learning, particularly in the space of machine learning, it gives an opportunity to try things out easily without having to know a huge amount of background. It's also nice because it has a lot of video support. It has a community where you can ask questions. They do run courses, and I believe from memory there is a textbook as well, an e-textbook available with this through the website. So hopefully that gives an overview of machine learning, how we can use it. I've shown you how not to use it perhaps on bad data, but that data was generated somewhat artificially. I'd be happy to answer any questions. We've got about 10 minutes for questions, and thank you for your attention.
Yeah, Graham, thanks so much for wonderful session and very nice demonstrations. So I think one thing which I liked is that even a lot of biologists who are here in this session, they can easily do this without going through much a lot of programming with Python or R. So they can still go through with Orange and try to do that. Question is that totally freely available, or we have to take some license version to access all the features?
No, everything is completely open source and freely available. So everything I've shown you is just what I've downloaded, and I haven't paid a penny for any of that. So,
so in fact, at least with students with my group, I would like them to start using it. And maybe I request you, Professor Graham, if you can give some assignment based on this, which we can ask them, and they have to then try to reproduce that. That can be a good way of knowing people have learned that. So we are recording the session anyways.
Where it's not quite so strong is probably on the big data. But what I've showed is the basics, but there may be ways of, for example, pulling out individual files for each gene, say, or protein in a data set and running those to actually, or taking another approach that said take a small set of features from the bigger set and run with those as well. Right. I believe Deep has got a question.
Yeah, go ahead.
Hello.
Hi, Deep.
Yeah, hi, sir. It was again a very good presentation. So yesterday, like last week, I was trying Orange, and I could easily, run with my data. I could easily run the SVM model and some of the machine learning models. But unfortunately, there is a place where I'm failing. I'm getting error. Is if you see in the widget, you will find a bioinformatics. And in the bioinformatics, there is I think non-parametric test is there.
I'm just looking now at the widget.
Yeah.
So
the bioinformatics widget is there.
I'll just share my screen.
Yeah. Yes.
Okay, so yeah, the bioinformatics widget.
Right, sir. So in that particular, there is a differential expression you can see, right? The kind of a Gaussian distribution. Yeah.
Yes. Yeah.
So if what I was doing, I was selecting the column and trying to do the. So if you select here scoring method, the drop down yes, this one. The scoring method. Yeah. So here there is Man-Whitney U-test is there, right? Which is non-parametric. So, but I was trying to do it, but somehow I was getting multiple like errors, and it was not happening. And I was using P cross N matrix only, the groups where I have selected the columns also. And on the same data sets, the other machine learning, all the like the random forest and other widgets were working fine. But this was not not at all working. I tried to see the help also, but somehow I couldn't complete that. I was getting errors. So so if you could help me, then I can share you one of the files, and that is where I was failing. It's a small thing, but still I was not able to do that.
Yeah. So I mean, I don't tend to use this software for pre-processing. I would probably use another statistical package.
Okay.
But if you send me what the output was, I can have a look and see what the error might be. Often the errors that I tend to find creep in are usually with the way the data is presented to the widget. So, and it might be that the best way would be to contact them and just see if you're presenting the data in the correct way or if there are any known bugs in that module. But the other way of doing it, I guess, would be to do it in a spreadsheet type sort of package, or to do it in R, or to do it in Python with the right module. But for this, I can see why you're using a Man-Whitney U, because it's non-parametric. But I guess there is. I just need to understand a little bit more about how, whether you were taking the data straight from Geo into it, or whether you were putting a separate file into it.
No. So I was not using a Geo over here. I was just taking one proteomics data set only. If you remember, we were trying to include both statistical approach and also the ML approach to do a ranking feature. So same thing I was trying to do it here also. So I was feeling if I already have I can make a network which will be having the statistical output also, and the same way I can use few of the ML widgets, and from both the output I will do a ranking method. But there only I was failing in the non-parametric analysis.
So I've just tried it for this data. Okay. Yeah, it didn't work for this data because no 0-6 scores undefined, 0 selected genes. So it didn't work for that, but it did work in terms of it ran the process. I'm just trying to work out how it receives data. Let's have a look. I guess we can perhaps discuss this offline.
Yes,
have a meeting next week. Just looking at, there's a lot of help here, but it might be that if you go to the Orange forums or fora as well, then it might be there you can find a way of an answer to it. Did the other methods work? So did the other approaches such as t-test or whatever work? Or was
no, sir, no. In the differential expression widget, nothing was working. I tried even the log2 fold chain also. I was trying. And I was trying to make a scatter plot over there after the correlation, but nothing was. Even the volcano plots I was trying. Nothing was working actually. So, but the other widgets are pretty good. I checked 2-3 widgets, especially the visualization and the model, and all the widgets were working fine.
Okay, so it might be, to me that suggests it's either the format of the data.
Okay, sir.
Missing in the format of the data that it's not recognizing. Did you use it for selected columns, or did you use it for?
No. I used for selected columns only.
Yeah, so you selected some sort of target class.
Yes, sir. Yeah. So I used PC over there, and from there I chosen some components, and in those components I was using. I was just playing with the parameters, but it was not working.
Okay, so so had you done a PCA on it? Is that what you're saying?
Yeah. Yeah, I have done the PCA, and like the way you were showing, you have taken 4 components and you have moved on. So the same way I was using 15 to 20 components, and I was trying to see how the scatter plot is showing.
Right, it might be by using the principal components analysis, you've collapsed the variance of the data, and so for some reason the differential expression might not be able to cope with the principal components.
No, sir, but I didn't link the PCA with the non-parametric. I linked the select data with the non-parametric.
I see.
Yeah.
I was going to say we'll talk about it offline and have a look into it,
Okay,
but it might be
Sure.
also worth speaking to Orange and just having a word with them, because they're usually quite good at communicating. So.
Okay, sir, I will do it.
Okay. Okay. Are there any other questions?
Yeah, Jyotirmay, go ahead.
Sir, for the stacking, we are taking the inputs from all the models, right?
Yes. Yeah.
So you told, suppose there are 5 models, and out of them, if we are taking the majority of votes, suppose 4 out of 5 gives them majority one. That's what we take in stacking, right?
Yeah. So what stacking does is it looks at the output of each of the methods,
Yes.
or each of the models, and then combines them together to see if we get a better performance.
Now, what I wanted to ask is it necessary for taking all the models into stacking? Suppose
No,
there are 5.
no. So what we could do is what we feed to stack, we could, let's say we want to remove naive Bayes. So if I just delete that, I'll just show, make sure I wasn't showing my screen. Right, yeah, so bear with me. Screen 1, there we go. So if you can see what I've fed into the stacking, so while I wasn't sharing my screen, I deleted one of the links, and maybe I want to delete that link. So now the stacking only receives information from 3. And so I can see whether that, well, that looks exceptionally good. But I think it's probably because I've done something to the data. But you can see I can select what goes into stacking. And I could theoretically, if I go to, is it evaluate, visualize? No, it's model. So if I take another stacking, let's see if this works, and maybe learner, okay, and learner, okay, and I put those into test and score. Yep, that's worked. So now you can see I've got 2 stacks, but those stacks receive different information from different learners and different machine learning algorithms. Notice there's perhaps a slight bug in here in that it doesn't name the stack. But if let's see if I can rename this. Rename stack. I'll call it STK2. Okay, does that update? No. Okay, so that's actually a slight bug in their system, in that by renaming it, it doesn't reflect that in the name of the list. But if I took one out, for example, I can see I've got 1 stack, that gives me 1. If I add that one back in and take that one out, oops, still running. Then I've got now the 1 stack in there gives me that performance as well. But for some reason, the name name of the stack doesn't track through to the test and score. But yes, you can put whatever you like into the stack.
Also, for the random forest model, on what basis do we select the number of trees and the depth of the trees?
So for the random forest, then again, there is no right and wrong answer for that. I think that it's a fine line, and again, it might be worthwhile experimenting with that. But generally speaking, we want to keep some level of simplicity in the tree, because if we have too complex a tree, then we end up with a very non-reproducible tree and a tree that doesn't produce generalized results. So what I would say is initially let the algorithm decide what sort of depth and number of branches, because there is some element of that in there, but also then experiment with increasing and decreasing the number of branches and the depth of the tree. But the problem is that if we increase the depth too much, then every individual case has its own large branch, and that doesn't give us generality. Whereas, so the idea really is to keep the number of branches minimal, but while maintaining maximal performance.
Okay, sir. Thank you.
Because a simple tree is easier to interpret, and there's all it's talking about generalization, the ability to predict for unseen cases. Well, a simpler tree is easier to generalize than a more complicated tree.
All right, any more questions or any comments for Professor Graham? Okay, if not, Graham, thank you so much once again for the wonderful lecture and
Pleasure.
very informative session. I'm sure many of the students and participants, they will take some of the knowledge back from here and they will start using these tools. And we are, of course, there and helpful to provide them more data if they want to practice further. So
Yep, I'm happy